The overall goal of this procedure is to clone and express antibodies produced by B cells. This is accomplished by first isolating B cells from a relevant source tissue, like blood or thymus. The second step is to perform FACS and select the B cell population positive for IgG production. Next, the cells are immortalized by infection with Epstein-Barr virus together with Toll-like receptor 9 activation by CpG-ODN 2006 and kept in culture. The final steps are to isolate RNA from clones that are tested positive for IgG production by ELISA and to generate copy DNA from both light and heavy IgG chains. Ultimately, the IgG sequences can be cloned in mammalian expression vectors to produce high quantities of monoclonal human IgG.
The main advantage of this technique over existing methods like phage display technology is that original IgG heavy and light chain pairing is not lost.
This method can answer key questions in the field of autoimmunity, such as discovery and functional characterization of pathogenic autoantibodies.
Don't forget that working with patient material and EBV virus can be risky. EBV virus is a human pathogen, and precautions such as BSL-2 procedures and wearing adequate protection should always be taken while performing these steps.
Before staining the PBMCs, plate them in a 25 cm flask with 6 ml of complete rPMI 1640 medium and let them recover overnight. After blocking and washing the cells, collect the PBMCs in a tube with a ½ milliliter of medium and spin them down. Then resuspend the cells in labeling buffer and count them. Next, prepare 2 FACS tubes of 5 million cells in 200 microliters of buffer. To these tubes, add 10 microliters of anti-CD22 PERCP or 40 microliters of anti-IgG PE and let them incubate for 30 minutes on ice and in the dark. Then wash, filter, and sort the cells. The goal is to count the double positive cells. Begin with diluting the sorted PBMCs 1 cell per microliter in rPMI 1640 complete medium with addition of CpG 2006 and EBV. Then, in a hood, seed 110 microliters of the cell dilution with EBV and CpG 2006 to wells of a 96-well round bottom plate, previously plated with WI38 irradiated cells.
The infection of 50 cells per well is optimized and increases the likelihood to obtain a monoclonal antibody. However, we recommend to confirm this by a PCR.
After a week of incubation, inspect the cells under a microscope for B cell growth. After 2 weeks, change the media. Slowly aspirate 90 microliters of medium from each well and transfer it to a new plate to test for IgG production by ELISA. Then, add back 100 microliters of fresh supplemented medium, including IL-2, to each well. Continue monitoring for clone growth through their 3rd and 4th week of growth, repeating the media change each week. See the text protocol for clonal expansion methods. Check the supernatant from the medium changes for IgG production using an enzyme-linked immunosorbent assay, or ELISA. First, prepare the ELISA plates. To each well, dispense 50 microliters of goat anti-human Fc antibodies diluted 1 to 200 in coating buffer. After incubating and washing the plate, block the wells with 100 microliters of blocking buffer per well and incubate the plate again. After the block incubation, knock the liquid out of the plate onto a paper towel. With the plate prepared, dilute the supernatants with 4 volumes of rPMI. Then, for standard controls, make a serial dilution of human IgG solution in rPMI from 1,000 nanograms per milliliter to 15.6 nanograms per milliliter. Also include a blank. Apply 50 microliters of the supernatant or standard to each well, and incubate the ELISA plate for an hour at 37 degrees Celsius. In the same manner, additional ELISAs can be set up to analyze further dilutions of the supernatants to test the antibody affinity. After the incubation, wash the plate. Then, to each well, apply 50 microliters of incubation buffer with peroxidase conjugated Fab2. After another hour of incubation and another wash, add 100 microliters of substrate solution with TMB to each well. Then, incubate the plate up to 10 minutes until the blue that appears stabilizes. Then, stop the reaction and measure the absorption. Any cloned supernatants with measured absorbance 3 standard deviations over the blank are positive for human IgG antibodies. Antigenic specificity can be detected by further assays. To begin, follow the text protocol to synthesize copy DNA from the positive clones. Then, set up PCRs for the IgG heavy, kappa, and lambda chain. In the results, find the heavy chain at 400 to 550 base pairs. Find the light chain at 300 to 400 base pairs. If they aren't there, use a microliter of products as a template for another PCR. The new products should be in the appropriate range. Then, proceed with cloning the best products. First, sequence them. Their electropherograms should be clean and correspond to a single immunoglobulin sequence. Following the text protocol, clone the purified PCR product into a microgram of plasmid DNA. Be sure to ligate overnight at 16 degrees Celsius using T4 DNA ligase. Prepare the hex cell in supplemented DMEM and grow them to 75% confluency in 75 square centimeter culture flasks. For each heavy, light chain transfection, prepare a mix with 1.75 milliliters of DMEM without serum. Include 4.5 micrograms of heavy chain vector, 3 micrograms of light chain vector, and 50 microliters of polyethylamine solution. Gently add this mixture to a plate of hex cells and swirl the solution over the plate. Incubate the plate overnight, and then begin collecting the culture media every day, replacing it with serum-less medium. The media contains the antibodies, and it can be collected daily for 4 days. After staining CD22 and IgG positive, PBMCs were sorted by FACS. The area of the double positive cells, B cells producing IgG antibodies, was collected separately for expansion. About 1% of the PBMCs were in this double positive population. After 5 weeks of EBV immortalization and CpG 2006 stimuli, immortalized clones were isolated. WI38 feeder cells have a more elongated fibroblast shape, and the B cell clones appear as very small round cells growing clustered in the middle of the round bottom multi-well plate. The supernatant of the growing clones was tested using an ELISA to IgG. A positive clone was evaluated on one with an ELISA value 3 standard deviations over the blank value. At least 3 supernatants from a positive clone tested positive in the ELISA. Antibodies from a positive clone were sequenced after cloning the immunoglobulin heavy and lambda chain pair. Variable and constant regions of the heavy and light chain could be characterized.
Following this procedure, other methods like sequencing can be performed in order to answer additional questions like antibody gene usage, genetic diversity, etc.
After this development, this technique paved the way for researchers in the field of autoimmunity to explore disease pathology and relevant therapeutic strategies for autoimmune diseases like myasthenia gravis.
The implications of this technique extend toward diagnosis and therapy of diseases like myasthenia gravis, because discovery of pathogenic autoantibodies can lead to the development of new treatments such as competitor antibodies lacking inflammatory activity.